So the kingdom Animalia. Let's first review some things about animal cells, just some of the differences between plants and fungi. You might recall that animal cells do not have a cell wall, and they have a cell membrane that has a glycocalyx, or that is protein filaments and collagen that is extending out from the proteins that are embedded in the membrane. So this is just a picture of the membrane there. And they also have remember they have to connect the cells because they don't have cell walls, and they don't, multicellular animals have to have the cells attached to each other. They have these different types of connections. The thing called gap junctions allows cytoplasmic connection between cells. This is homologous with the plasmodesmata of plants, allowing cell-to-cell communication. They also have something called a desmosome, and desmosomes are kind of big junctures. And then they have something called tight junctions, and tight junctions are basically kind of like little rivets and so forth that sort of hold the cells together. So those are they are also heterotrophic by ingestion. This is different than fungi because fungi are heterotrophic by absorption, and we've been talking about that with the fungi. And of course, plants are autotrophic. And they are multicellular eukaryotes, which is not different than plants or fungi. That's a similar thing. Though one of the big things, though, is they are motile, meaning that they move around. And they also, most of them do anyway, or have part of their life cycle that does. And they have sexual reproduction, where the 2N or the diploid stage is the dominant part of the life cycle. They do not have alternation of generations because they don't have a multicellular gametophyte or 1N stage. They go through a series of embryonic development that is kind of unique. They start with a zygote. The zygote, of course, is created from egg and sperm, and it is one cell. It will undergo mitosis, and it will do that for a bit. This is showing the 8-cell stage. These have some different arrangements we're going to talk about later, separating the different animal groups. And then it'll do something called cleavage, and cleavage is basically where it forms a blastula. That is, it forms a ball around, or like a hollow ball of cells. So instead of being completely like a mass of cells, it's a ball of cells. And then it can undergo something called gastrulation to make a gastrulo. And what that means is it can push. It's kind of like sticking your thumb in a wad of clay that is like a ball of clay, and put your thumb inside of it, and that's pushing through, and that's going to end up making the gut cavity. And at first, and depending on which end it forms, it's going to separate out some of our different groups. And you can see some of the names to this, and we will go over this a little bit more later. All right. So they also have Hox genes that are similar for all animals. And you might remember in Core 1 talking about this, the Hox genes being common to all animals. Even the lower groups of animals have a couple of sets that are common to the higher groups of animals. And then as we go through the animal groups, everything that is, I think it's above fish or chondrichthyes maybe, everything above that has 4 sets, 4 sets of 8 Hox genes. And those are, Hox genes are control genes that control body form. So they control the development of a particular body segment. And you're going to see that segmentation is one of the features that develops through the animal groups. Of course, just like plants, we are going to be looking at the groups from water to land. You're going to see the features developing as organisms move onto land. So let's just talk a little bit about the evolutionary history. Now, this is not at all complete, And I want you to read that section. But some of the major points of this are, first of all, to know. Let's go over first of all. I messed that up. First of all, 99% of the species that have existed are extinct now, and they have diverged from fungi. And remember, we talked about when we were talking about the fungi, we were talking about a common ancestor called choanocytes or choanoflagellates. And they have these cells that they're made of, like here's the cell body with little extensions off the cells and a flagella. So this is kind of like a feeding cell. And you're going to see that in animals, some of the lower groups, that is sponges, are going to have a cell that looks just like that. So this is one of the things that lends itself to the concept that these are related. All right, so they diverged from fungi about 1.2 billion years ago. And throughout the history, of course, like when things were changing, organisms going up and down, but during what they call the Cambrian explosion, which is 542 to 251 million years ago, it brought about huge changes in the Hox genes. In other words, they thought conditions were probably changing to the point where the Hox genes were changing sort of regularly. It may have been perhaps more radiation, the formation of the atmosphere as we know it today. Who knows exactly what was going on that was causing that, but there was huge changes in body form during the Cambrian explosion, and because of that, there was also a lot of ecological niches that changed, especially in regards to predator-prey relationships. Predators being things hunting and prey being things being hunted. Things, like mimicry, defense mechanisms, ways to escape your predator, ways to get your prey. And the movement of organisms became more pronounced, and at the same time, oxygen levels were rising, land masses were starting to move about. That meant that some of the land masses that were heretofore, like in colder climates, became in temperate climates. Plants invaded land, so you had more oxygen in the atmosphere, and this enabled more motile or more movement in animals, more active organisms because of the oxygen. All right, so and then there were several mass extinctions over that period of time, and leaving fish or vertebrates, fish on top, and they think that a tetrapod, that is a four-legged organism that was kind of derived from the fish, went onto land about 360 million years ago, creating amphibians first, that are organisms that kind of need water in their life cycle, frogs, salamanders, toads, etc. And then creating amniotes. These are things that either have an amniotic egg or have internal development of the young. And the amniotic egg is kind of like a enclosed embryonic sac, so to speak. And reptiles and mammals have that, of course. And then it was about 65 million years ago, 66 million years ago, that insects really started to increase, and so did flowering plants. So things were kind of taking over the land masses, and insects, as they went up, they're developing relationships with flowering plants in terms of pollination and etc.